This is a video to help you through the activity on viruses and population evolution. Viruses are tiny packets of nucleic acid surrounded by a protein envelope, or a protein coat. The two features that are common to all viruses is that they contain nucleic acid surrounded by protein. In your textbook, this figure shows the steps of the viral life cycle. There is a lot of debate about whether viruses are living or non-living. They share features of living organisms in the fact that they reproduce. However, they are not autonomous. They can't reproduce by themselves, and they need machinery that's present inside host cells in order to replicate. And this is why many people argue that viruses are non-living, because they do not contain the pieces that are necessary for replication. For a virus to infect a host, it first needs to attach to the cell surface and inject its DNA or RNA into the cell. The DNA and RNA will then be used to replicate the viral nucleic acids and to make new viral proteins. The nucleic acids and proteins will then be assembled into new virus particles that will bud off or leave the cell in order to infect new cells. I'm going to talk about 3 types of viruses in this activity. Your textbook only talks about 2 of these. So please note that there is a discrepancy between this information and the information in your textbook. The first type of virus that I'd like to discuss is a DNA virus. A DNA virus is characterized as such because the nucleic acid inside its protein coat is DNA. So DNA viruses contain DNA nucleic acid. The first step in the viral life cycle is that the virus must attach to a cell and inject its DNA into the cellular cytoplasm. Viral DNA cannot be replicated in the cytoplasm, and so it must enter the nucleus. Once inside the nucleus, the viral DNA is transcribed by the cell's own RNA polymerase into viral mRNA. In this process, the virus hijacks the cell's RNA polymerase. The viral mRNA will then move out of the nucleus and will be translated by the cell's own ribosomes. This translation results in viral protein. While this is happening, the viral DNA in the nucleus will also be replicated using the cell's own replication machinery. This DNA will also leave the nucleus. Viral protein and viral DNA are then assembled into new viral particles, which can then bud off and go on to infect other cells. The main features of a DNA virus are that it contains DNA and that the DNA must get into the nucleus in order to replicate virus. Now let's talk about a regular RNA virus. We'll see 2 types of RNA virus, the retrovirus and the regular RNA virus. The main difference between an RNA virus and a DNA virus is the type of nucleic acid that is in the virus. RNA virus contains RNA. Viral RNA is injected into the cell. In the cytoplasm, the RNA can be directly translated into viral protein. A main feature that characterizes an RNA virus as a regular RNA virus is the production of the protein RNA replicase. RNA replicase is used in the cytoplasm to replicate viral RNA. After viral RNA and viral protein have been produced, these components will be assembled into new viral particles, which will then bud off and infect other cells. The main features of an RNA virus are that it contains RNA rather than DNA, and a regular RNA virus will contain the RNA to translate RNA replicase. Regular RNA viruses do not have to enter the nucleus in order to replicate. Now let's talk about RNA retroviruses. RNA retroviruses also contain RNA, and they inject viral RNA into the cell cytoplasm. In the cytoplasm, the RNA is directly translated into viral protein. The main difference between RNA retrovirus and a regular RNA virus is the type of proteins that are produced at this step. An RNA retrovirus will make reverse transcriptase and integrase. These are 2 enzymes that allow this viral RNA to integrate its own DNA into the host genome. In order for this virus to replicate, the RNA enters the nucleus, along with the protein reverse transcriptase. In the nucleus, the reverse transcriptase reverse transcribes the viral RNA. This is the opposite of transcription. Transcription takes DNA and makes RNA. Reverse transcription takes RNA and makes new viral DNA. Once the viral DNA has been synthesized, the enzyme integrase, which was also produced by the virus, integrates the viral DNA into the host genome. Now the viral DNA is a permanent part of the genome of this cell. Once this happens, regular transcription and translation can happen, along with all of the other host proteins. Viral RNA and viral protein are then assembled into new virus, which can go off and infect other cells. Retrovirus infections are particularly difficult to get rid of because they integrate the viral DNA into the host genome. These viruses are also particularly prone to mutation because the reverse transcription process is not a high-fidelity process, which means that many errors occur, which means many mutations can occur. These viruses often mutate quickly and integrate themselves into the host genome. Now I'd like to talk about antiviral strategies. One antiviral strategy is the use of vaccines, or the use of our adaptive immunity. Adaptive immunity is the production of specific antibodies by cells that are designed to attack a certain type of infection. The production of specific antibodies involves complex cooperation between immune cells. First, the macrophage traps virus particles. It processes these and becomes an antigen-presenting cell. The antigen-presenting cell then presents the processed virus particles to helper T cells by direct cellular contact. The helper T cells become activated and undergo clonal expansion. This means that it makes more of itself, memory helper T cells that will remember the virus target, and causes the B cells to become a plasma cell that makes the antibody against the virus. The antibodies attach to the virus and destroy it. The B cell will also clonally express to make memory B cells that will remember which antibody to make against this virus. Memory cells wait for the future invasion by this particular virus and respond quickly. This memory response is how vaccines work to target viruses, and also why we usually do not get the same cold or flu twice. The cartoon showing how specific antibodies are made demonstrates how vaccines work too. The vaccines are made from killed virus or viral proteins that are made in a lab. These viral proteins are injected into the body so that your immune system can start making antibodies against them. Several immune cells work together to create the memory response that results in specific antibodies. That way, if you are infected with the real virus, your immune system will be ready to respond. The best part about adaptive immunity is that every one of your cells that responds to the initial virus will make its own antigens and own antibodies that will be slightly different from one another. And every person's immune system is different. So every person will have several different antibodies against the same virus. It's very difficult for viruses to adapt quickly enough to overcome this system. And so it's harder for viruses to overcome or mutate to become resistant against these mechanisms. The other antiviral strategy is antiviral drugs. Antiviral drugs are not comprehensive strategies, as the adaptive immunity is. Adaptive immunity attacks viruses from several different directions, or attacks several of their proteins at once, whereas a drug can only inhibit one enzyme or stop one process at once. The problem with antiviral drugs is that viruses can become immune to them, or they can develop antiviral resistance. Let's talk about one of the strategies that doctors use to fight HIV. HIV is a retrovirus. Here are figures from your textbook that show the HIV replication cycle. These little X's over each step show where drugs have been developed to inhibit each of these processes. Each one of these processes is blocked by a different drug. Let's talk about the reverse transcriptase that is blocked by nucleoside analogs. Reverse transcriptase makes new DNA, and in the process of making new DNA, it must use deoxyribose nucleosides or nucleotides. This process can be blocked through a nucleoside analog like AZT. Here's the structure of AZT. Notice that it has a thymine here, and it's a deoxyribose analog, except instead of the alcohol group at the 3' position, it has an azide functional group. This azide functional group cannot be linked to another nucleotide. So when this is incorporated into a DNA strand, it ends at this 3' end. There's no way to connect this to another nucleoside or nucleotide, and so DNA replication is blocked. Nucleoside analogs like this block replication or production of DNA, and so they block reverse transcription of viral RNA into DNA. There are several other types of inhibitors that have been developed to block other points along the HIV replication cycle. When only one drug is used, it's like blocking a pathway. Since this is a retrovirus, the virus can quickly mutate to overcome the blocked pathway, or in other words, it can find a way around. For example, this is essentially a substrate for the enzyme reverse transcriptase. Since enzyme active sites are very specific for their substrates, some mutations of the reverse transcriptase protein could result in recognition of this particular type of nucleoside analog. If the viral protein learns to recognize this, it will no longer use this, and it will be able to recognize this versus a regular nucleoside. If a virus would mutate and start producing a protein that could select for a regular nucleoside over this nucleoside analog, the virus would have adapted antiviral resistance to this particular drug. To avoid antiviral resistance, many doctors use cocktails of antiviral drugs. For example, they might use an integrase inhibitor as well as a nucleoside analog, as well as a protease inhibitor, to hit the virus at several pathways, to make it less likely that the virus will mutate and overcome each of these obstacles. Now let's talk about H1N1 and Tamiflu. H1N1 is what we know as swine flu. It's an RNA virus. Tamiflu was a drug that was developed to stop this virus. Here's a picture of H1N1. Inside of the protein coat, you see RNA. The protein coat surrounds the RNA, and there are specific receptors on the surface of the protein coat. One of the proteins on the cell surface is neuraminidase. Here's the scheme for replication of an RNA virus. And what Tamiflu does is it blocks virus exit. Here's a picture showing how a virus, after it's been replicated, is released from the cell membrane. Once it's on the outside of the cell, there's a cell receptor on the membrane that prevents it from leaving. The viral protein neuraminidase actually cleaves this receptor and releases the virus so that it can go infect other cells. A neuraminidase inhibitor like Tamiflu actually blocks these neuramidases from working. And so now the neuraminidase cannot cleave the receptor. So the virus is stuck on the cell membrane and it can't go infect other cells. Tamiflu is only inhibiting this one process, and viruses can quickly mutate. Flu virus has already learned to mutate so that the neuraminidase inhibitor does not block this activity anymore. Viruses acquire antiviral resistance through evolution. Here is a scheme from a scientific paper in Nature Reviews that shows how evolution happens. There are two examples of how this happens, but the main features are that there must be genetic mutation and there must be a stress. Evolution is the change in the distribution of population. Here's an example. If we have a population where there's only one individual that has a genetic mutation, and this genetic mutation happens to offer advantage to a stress, like an antiviral medicine, when the stress is applied, for example, an antiviral medicine, it will kill all of the non-resistant strains, or it will kill the regular individuals, and only the mutated individual will survive if that mutation offers an advantage. Once this is the only surviving individual, it will replicate and it will result in a population now that has a very different distribution than the original population. Now the entire population is resistant to drug or resistant to the stress. Then the next time you treat with the same drug, none of the virus is killed or none of the individuals is killed because all of the individuals are resistant. This is one way that evolution can result from one genetic mutated individual. Another is when you have many mutants in a population and they all have different levels of resistance. Here, the non-resistant individuals are the lightest color and the most resistant individuals are the darker colors. When you introduce a drug here, you kill the individuals that are not resistant. These individuals can mutate further and replicate to make more of the resistant individuals. And if genetic mutation is common, then further genetic mutation can lead to even more resistance. Then the next time you add the drug, you've selected for the most resistant individuals. The population changes because of mutation and because of stress. In this case, the stress is a drug. Thanks for watching this video on viruses and population evolution. If you have any questions about this video, feel free to post them in the comments. And if this video was helpful for your learning, please let me know by giving me a vote.